Hello and welcome everyone. My name is Anastasia Ryasanova. I'm a coordinator of the Treesearch research infrastructure. And together with Gunnar Westman, we would like to warmly welcome you to the last webinar within the NMR topic. And it's a great pleasure for us to welcome adjunct professor Tomas Larsson from RISE and KTH, who will introduce us to the field on the NMR topic on the isolated cellulose. So please, Tomas, the floor is yours.
Thank you. Thank you, Anastasia. I'm happy to be here, and I hope that I will give you an interesting hour. I will talk about an hour about carbon-13 NMR on isolated cellulose, and actually it's in solid state, so it's CP mass carbon-13 NMR on isolated cellulose. So I'll roughly talk about this. We will talk about how you can use this technique or this tool to have a look at cellulose, isolated cellulose and cellulose-rich pulps. And then I would like to say a couple of words of cellulose, because when you start to extract information about cellulose with either technique you use, you end up in sort of tricky area of interpretation and understanding. So I would like to share that as well. And then you can, NMR doesn't stand for itself. You can compare it and use other techniques to obtain complementary information. So I'll talk a bit about that. We also would like to use a technique called FSP and NMR to address, well, not only what's there, but what's not there. With that, I mean porous materials, because when you isolate cellulose, you normally end up with a fiber wall, and that can be quite porous. And pore sizes are of importance as well as particle sizes. And then last but not least, then a way forward, and I'd say a few words about some test runs we have made with the new NMR, solid state NMR technique, which is cryomass, where you use a cryo probe, not only for solution state, but also for solid state. So, and I will talk for about 1 hour. So if you have questions or comments, feel free to interrupt me at any point, and you can raise your hand and make Anastasia interrupt me. But with that said, I have lots of ground to cover, so let's start. So I start with this, and this is cross-polarization, magic angle spinning, carbon-13 nuclear magnetic resonance spectroscopy. And we look on isolated cellulose and cellulose-rich pulps. And we want to have information about the supramolecular structure. And if you look at celluloses isolated from different sources in nature, you get spectra that look something like this. And these are all recorded in the water swollen state. And it's obvious to the naked eye that there are differences between these spectra. And in brief, if I should turn on my pointer just a minute. Groups like this that come from carbon atom 1, 4, 2, 3, 5, and C6 in the anhydrous glucose unit, they are given, the position of the groups are given essentially by polymer or molecular configuration, how things are attached covalently. And the fine structure within a group is given by polymer conformation. So that's the information we seek, and they look different, although all of these materials are made of the same polymer. So obviously it's about how polymers are packed together that gives these differences in these in these spectra. And that's the supramolecular structure, and that's what we were looking to obtain information about. So we take that a step further by using the region where signals from the carbon atom 4, the one that is next to the glycosidic linkage, shows up, because that happens to be the region that has the highest apparent resolution. And you can put a couple of things together here in order to obtain information about the supramolecular structure. First of all, you need to have some kind of idea about the model, the physical model of the material. And that is a fibril, that's the square cross-section here, and then there's a fibril aggregate. And this is just a model of a 2x2 fibril aggregate. But all the different states of polymer order that you can sort of identify from a model like this can be found as separate signals when you do the proper fitting of this region of spectra. And you can see that we have the envelope of the fibril aggregate, which is in contact with surrounding liquid. And then you have the interior surfaces in the fibril, which is also surfaces, but they are mainly contact between fibrils. And then you have the crystalline interior, and you have a transition zone from the surfaces to the interior. And all of these can be identified as different separate signals showing up in the interpretation we use. There are a couple of things to say about this. First of all, you need two different line shapes, and the crystalline signals, the gray one that represents signals from carbon atoms in a very highly ordered state, or the crystalline state, they are of a Lorentzian type. And the other ones, the blue and red and purple and so on, they are of Gaussian type. And that comes from the fact that they are of less order. So they are not as highly ordered as the crystalline polymers in the interior, in the crystalline interior. There are a couple of things to say about this. And first of all, and I come back to this, I won't talk about all these things in this particular slide. Well, I come back to that. We have two surface signals, and that was a bit of a riddle. We have a very broad signal that comes from the interior surfaces where fibrils are more or less in contact. But we can, based on this part of spectra, establish experimentally a surface to volume ratio, or the total number of polymers in a cross section compared to the polymers around the perimeter of the cross section, whether it's for fibrils or the fibril aggregate. And since a polymer has a pretty much constant thickness, we can take measures of the lateral size of fibrils and fibril aggregates, as long as we have a cross-sectional shape, which we assume to be a square. Not only that, the polymers that constitute the surface of a fibril, they are separate signals over here, and the rest of the polymer signals are over here. This is from the C4. And this means that there is a difference in the position of the C4 for surface polymers compared to polymers in the interior. And that means in our vocabulary that the polymers on surfaces have different conformations compared to the interior. But the interior is classified as a lattice of cellulose 1. It's a lattice type that has certain characteristics according to XRD. And that means that if a polymer doesn't have that conformation that is characteristic of the cellulose 1 lattice, then it's not a cellulose 1 lattice. And we can see that surface polymers are distinct. There are about 4 or 5 ppm in carbon 13 shift different due to difference in conformation. So for that reason, we say that the surface polymers are not crystalline, and we are considering those to be non-crystalline, not amorphous. They have a rather high degree of order along the fiber, but they are not crystalline. And based on that, we can determine the degree of crystallinity based on a NMR spectra like this. And that is effectively also related to the surface to the volume ratio, because the number of polymers sitting on the surface, that's basically the fraction of the material that is not crystalline. So we can get dimensions, lateral dimensions, and degree of crystallinity from just doing the spectral fitting this way that we have shown here. But doing spectral fitting it builds on one assumption, and that is that all these fitted functions and the signals that you record, of course, are representative of amount in sample with the same, if you wish, response factor. And to figure that out, we have established that this turns out to be the case for cellulose. With an internal standard, we could determine gravimetrically how much polyethylene and cellulose we had in a sample. And then we determined that this is the gravimetrical determination is the horizontal line. And the markers here, they are the experimental measurement that we obtained by spectral integration. And now there is in the measurement sequence of CP-mass, there is one crucial parameter which is called the contact time. And you can see that the important thing here is not to keep the contact time too short. If you're above some 600 microseconds, the gravimetrically composition and the composition that you obtain from integration of spectra, they agree. And that means that we have a quantitative spectra. everything we put into our sample holder when we do measurements shows up as signals intensity in the spectra, and that's something you need to know. Secondly, since we are dealing with the surface to volume ratio, it could be also be nice to have some kind of proof or experimental support for the fact that the signals we think are surface signals indeed are. And one way to do that in a rather direct fashion is to just replace water with another liquid, which in case was methanol. And you can see that the signals that suffers the most when you do that replacement are, of course, the signals in direct contact with the liquid, which are the surfaces. So that's also a nice piece of experimental support for this interpretation. We went even further and also measured relaxation time on cellulose, and this is rather time consuming in the solid state, but it's important because the relaxation time, for those not familiar with NMR, is essentially during a measurement you push energy into the spin system, and the time it takes for that energy to come out of the spin system and letting the spin system go back to equilibrium is the spin lattice relaxation time. And we have measured that, and from a practical point of view, we can only determine the relaxation time for a couple of signals, the least ordered ones, or the surface signals and so on. In the crystalline interior, the relaxation times or spin lattice relaxation times, they are so long so we can't measure them from a practical point of view. But we could determine two different relaxation times for the two different surface signals. And I remember, if you remember, these are the signals that comes from the envelope of the fibril aggregate, or from the the different fibril surfaces, so to speak. So that's interesting, and it's also an indication that supports the interpretation we use. And the model, the fibril model, this all boils down to is the slanting ribbons here are, of course, the simplified polymers, is this. And I'll come back to this a couple of times during this presentation, but this is the typical fibril that you get when you isolate cellulose from wood. It's around 4 nanometers wide. It has a reasonably square cross section. It's not exactly, but roughly square cross section. And it has two pairs of phase boundaries. You have surface polymers that resides on top of a crystallographic plane that's indexed as 1, -1, 0. And then you have another pair of surfaces that are surface polymers that reside on top of another plane that's 1, 1, 0, indexed as 1, 1, 0. And this is a very functional model that we have been using for some 25 years or more, I think. And so far, we have not been able, and nor has anybody else been able, to falsify this model as a interpretation tool for whatever measurement you do on cellulose. And it's, in that respect, very well established, and it's a very helpful and useful model. It is not, I should point out, it's not for all cellulose, but it's a good model for cellulose isolated from wood. Well, we use the C4, and the reason we use C4 is that that's actually the only region of the spectra where you can get access to all these different signals by spectral fitting. But if you could selectively label different carbon atoms in the anhydrous glucose unit, you could perform this spectral fitting on other regions of spectra as well. The problem is that typical plants, they get their carbon from carbon dioxide, so we can't selectively label anything. But bacterial cellulose use sugars as one of its carbon sources, so we can use selectively carbon-13 enriched sugars and feed them to bacteria, and that way build up bacterial cellulose that is selectively enriched in carbon-13. And you can see that we have, for reference, a couple of different celluloses, but they have bacterial cellulose, which is a rather highly crystalline one. But if you feed the bacteria selectively C4 enriched, you effectively get the spectrum that is only containing C4 because of the sort of acceleration that NMR gives you on concentration. So this is just 10% carbon-13 enriched, and still the spectra is dominated by the signal. You could do the same for C6. And once you have these three, spectra, you can of course do the spectral fitting. First of all, C4 in the native, C4 in the enriched, and you can also do on the C6, which has pretty much the same structure as the C4 with the crystalline interior and surface signals. And from that you can establish the surface to volume ratio, and you can compute, as we did, crystallinity. And you can see for all these three samples, we get pretty much the same degree of crystallinity, whichever region we use for determining it. And this has a kind of implication. It has an implication that says basically this, that any domain of polymer order, whether it's a surface or it's the crystalline interior, it's a paracrystalline or whatever, the typical domain size is large in some respect compared to the anhydroglucose unit. All the carbon atoms in one anhydroglucose unit sort of belongs to one kind of ordered state. And I think that's useful also to know for the interpretation of spectra. Just to show you why it's favorable to use water swollen samples when you record an NMR, carbon-13 NMR, you can see that we lose quite a lot of resolution when we have a dry sample. And this is not dried to an extreme, in an extreme sense. This is when you leave your material just lying around in ambient conditions. You might have some 6 to 7% or water in this, or moisture in this material, but still it's not sufficient to release the stress concentration that can be built up during drying. The surface regions, they are broadened and they lose their shape because we are now effectively constructing a very, very large fibril aggregate. So that's not so strange. But what is more interesting is that even the signals from the crystalline interior of the fibril get distorted and broadened, which means that we induce some kind of disorder in the dry state, also in the crystalline lattice. And you can see it both in the C4 and the C6. We have the same story here. And normally that is what we refer to as drying stresses. We build up drying stresses. We have geometrical mismatches that when water is removed, they don't fit quite well together, and for that reason we can get sort of tilted or twisted or bent or whatever crystalline domains, which is then becoming, in a sense, less crystalline. It's perfectly reversible after first time drying. You can go back and forth between these two spectra without much change. But after first time drying always induces a permanent or more or less irreversible change. So well, amorphous cellulose, I told you that the surface polymers are not amorphous. No, they're not. But you can, no doubt, you can make amorphous cellulose by just dissolution and reprecipitation or coagulation, whatever you want to call it. And you can see then that you don't get, in that it done this way, this is a gel-like cellulose bead that are only a few % of solid content, but you can see that the crystalline signals that come in this region, for this particular case I show cellulose 1, but cellulose 2 and 3 and 4, they all show up crystalline, they show signals in this region, and they are absent in this material. So here we have a truly amorphous cellulose, if you wish, but it's not the kind of polymer order we have in the surface polymers, because these signals are both at different positions and also less broadened. And the NMR technique has many benefits, and one of them is that since it's based on a surface to volume ratio, the surface signals becomes, of course, relatively larger when you decrease the size. So you can get very precise measurements of small structures. And it also means that the precision you get, we can talk about accuracy, but the precision you get is really high. And it's in that respect a good technique for tracking small changes. If you do some kind of processing of your cellulose material and you induce small changes in the supramolecular structure, this is really a good tool to track that, because you can get measures of these changes that are statistically significant because of the good precision. Now, I want to switch over to talk a bit about cellulose, because it's not just cellulose and it's not that straightforward actually. And I would like to start out with saying that, well, of course, cellulose 1, what we get from nature, you can isolate that from different sources. You can have from plants, algae, bacteria, etc., and animals and whatnot. But they are, of course, not the same. They don't have the same supramolecular structure. The supramolecular structure depends on the origin of the material. But not only that, even if you stick to a certain source, the cellulose supramolecular structure prior to isolation may have a different supramolecular structure compared to what it is after isolation. And this has actually caused much confusion. I'll come back to that. And if you take it even further, you can take the isolated cellulose and do chemical modification. And then once again, you can change the supramolecular structure. So none of these 4 different kinds of cellulose 1s, they are still cellulose 1 to a large extent, but they can still have very different supramolecular structure. And the one, if we look at cellulose before isolation, there has over the years, and I'm not trying to be funny now, but this is actually the situation. It started out a long time ago when everybody was pretty certain that the cellulose fiber should have 36 chains. And that's because in plants, you have this cellulose synthesizing complex that is a rosette of 6 units, where each of the 6 units can generate 6 polymers. So in all, that's 36. But as it turned out, it seemed that not all these 36 cellulose making or polymer synthesizing machineries were turned on at the same time. So then it was reduced to the 24 chain model, and that lived for quite a while. And then it was even taken further to what has been lately the most common idea about the biosynthesis of cellulose, which is the 18 chain model. I saw recently, beginning of this year, there was yet a paper that surfaced saying that, well, they have found the 24 chain model once again. But in many cases, these are ideas that are inferred from detailed studies of this cellulose synthesizing complex. And it's not measured. It's kind of difficult to measure this. I've seen a couple of X-ray measurement or papers where they have looked at cellulose nanofibrils in wood, that's prior to isolation, which has shown sizes that is close to this 18 chain model. But and in some cases, they have managed to isolate this cellulose synthesizing complex from the cell wall and kept it alive in a nutrient solution where it's been active and producing fibrils and so on. But it's tricky, but it might well be the case that immediately after biosynthesis, there are lots of publications on this, and there's no doubt about that they have been doing this in a serious fashion. So it might be correct that this is the latest suggestion from 2016. But you see that if you have an 18 chain model, you have a size, the largest dimension here is around 2.3 nanometers, something like that. And that might be okay. But originally, the statement about the rosette complex, this 6x6, you can actually see them here. These are the rosettes. You can see 1, 2, 3, 4, 5, 6, and still, even at this high resolution, you can't distinguish the interior of each of these cellulose synthesizing complexes. So there are actually believed to be 6 in each of these 6 groups. So for that reason. But one thing that is interesting to note here is that the size of this complex is about 25 nanometers. And that's about 10 times as large as the suspected fibril, which is the 18 chain 2.5 nanometer wide fibril. And now the polymer that's been made here, the beta-1,4-D glucan, is a rather rigid and stiff polymer. So when it's made and spun out of this complex, it will take quite a distance before these polymers can come together. And the consequence of this, I would say, is that it's not at all obvious that the shape of the machinery, which is a nice looking rosette, should be reflected in the structure that it makes. And I think this has been a paradigm for a long time, that the 36 chain model, for example, that should be hexagonal, but I don't see why that should be necessarily the case due to this difference in size. So, okay, and with that said, if you move on to isolated cellulose, and my focus has always been for a long time on cellulose isolated from wood, so I'll stick to that one. There are other sources as well, but plant cellulose, we can say. We are back at what NMR says mainly. So I showed you this one, and we have this region of spectra, and we have this model of a fibril, and it's of course not 4 nanometers. It's it's not 2.5 or 2.3 nanometers. When you have 1, 2, 3, 4, 5, 6, 7 chains, and they're a half a nanometers wide, or 0.6, you realize that this is a structure that's about 4 nanometer. And we never find the 18 chain fibrils after isolation. And it's not only us, there are quite a lot of other researchers that have shown the same thing, that the 18 chain model might be the fibril that you have prior to isolation, but after isolation, it's not. And exactly what happens and how this happens, how you move from an 18 chain fibril to a larger 4 nanometer, which is roughly 50 polymers chain model, we don't know. That's the honest thing. I can show you this one. This is from X-ray, a pretty good job from 2008, and you see that they are Avicell is isolated. It's a cellulose isolated from wood, that they come back with like roughly 4 nanometers wide as well. So there seemed to be a discrepancy, but I don't think this is so surprising, because if you take a piece of wood and put it in alkali and sulfidity at 150 or 160 degrees centigrade for several hours, why should the cellulose be the same after treatment? And you can see that other determinations made with AFM, with microscopy, you see that we get, for a rather mildly charged enzymatically degraded cellulose, you get 4.6, 5.5 nanometers. So it's just not a single time that this lack of 18 chain fibril shows up after isolation. It's more of its consequences so. And there has been ideas also about different shapes of fibril and different sizes. And this is from a rather recent paper in 2021 where they proposed this. And this is based on X-ray, but it's unfortunately based on X-ray alone, because if you take results from one technique, it can be, I would say that we need measurement results from several. And I mean, solid state NMR and cellulose has been done for quite a while, and there's no secret that we have 2 surface signals in solid state NMR. And we can explain that, and we have pretty good support experimentally for the fact that it comes from a shape that looks something like this. And if these 2 surfaces gives us distinct signal positions, if you were to have a third surface, it's rather unlikely that it would distribute its signal intensity evenly between these 2. Rather, we should have a third signal showing up, and we don't have that. So this idea with a hexagonal fibril that basically expose 3 kinds of surfaces is not really that well supported when you start to pool all kinds of measurement techniques together. You can see that we have, in this case, a surface to volume ratio that is roughly 50%, and it's the same thing you have in this structure. And I don't remember how many polymers exactly there are. Well, there are 36, but you have these 2 extra surfaces, which it's difficult to reconcile with what we know from solid state NMR. So that doesn't fit. This model, on the other hand, can be made consistent with both experimental results from NMR and from wide-angle X-ray scattering. So if you take it one step further, if you have isolated your cellulose and then start to do chemical modifications of it, then you can change it further, of course, because that's what the chemical modification do. And you can see in this case, we have changed the material by tempo oxidation. And we see that, well, tempo mainly affect surfaces. And we have, the idea is that tempo oxidation oxidize the hydroxymethyl group, and you get the primary alcohol turned into a hydroxyl or carboxylic acid. So C6 should suffer, as it does, but C4, at C4 you have an impact on the spectra also at C4, and this comes from the details of the material being a solid, and you have an impact on the electron structure if you alter the C6 position also on the C4. But it's quite obviously so that you have an impact on the supramolecular structure. If you do this, it's not the same material as you started with. So chemical modification changes things as well. And I have to bring wide-angle X-ray scattering into the picture now, because we start to compare now these two techniques, which are very good techniques for looking at supramolecular structure. So if you have seen wax on cellulose before, you know that we have mainly 4 signals. This particular measurement didn't reach up to the 004 signal at 35.2 theta, but most of the signals, they're around here, and they can be used to identify this as a cellulose 1 lattice. Now, if you do tempo oxidation, as we did in the previous slide on NMR, you can see that also here we have an impact that is in agreement with what we saw from the NMR. And we also change this material even according to wide-angle X-ray. They call it wide-angle X-ray diffraction, but it's the same thing. So that changes too. And you can see that they managed to take a fibril apart in this particular piece of work into strips that are of a width, a typical width as determined by AFM, I think. No, it's SEM, sorry. The width is around bit more than 4 nanometers. There's a distribution, of course, but also they can determine the thickness of these strips with AFM. And you get essentially a single molecular layer. You get a sheet of polymers according to the measurement of AFM. So, and obviously that's not the same supramolecular structure that you had in the starting material, or even after isolation. And you have seen this also by other microscopy techniques. You can see that when you increase the charge, you do carboxymethylation to higher and higher degree, you can also drop the fibril dimension down. The TEM, the cryo-TEM and AFM are not in absolute agreement here, but I would say that the trend is towards smaller structures. So it's still even further so. But this is a point worth keeping in mind, that first of all, if you are looking at cellulose, talking about cellulose, it's important to distinguish, first of all, what's the source, and then whether we are talking about cellulose in the living tissue prior to isolation, or if it's isolated cellulose, or if it's a chemically modified cellulose. And all of these three can have distinct supramolecular forms. There is absolutely no reason why they should be the same. So comparing NMR and X-ray, I go back to this idea of measuring relaxation times, and we can identify 2 relaxation times. Relaxation times, we can obtain them or estimates of them from molecular dynamic simulations. And we did that in order to figure out why there are 2 surface signals. And you can see that they are different, and that is a good thing when you want to compare hypotheses. So what we did was that we formulated 2 hypotheses that were popular at that time, and one of them was that we have 2 pairs of distinct surfaces. And it's not so far-fetched, because if you look at the best X-ray data there are structural differences between that surface or those 2 surfaces and those 2 surfaces. Not large, but there are differences. Packing distance between polymers are different, and some angles are different between the polymers along these lines, this side, sorry. And the other one is that if you look closely on a polymer packed together as a cellulose 1, you see that the hydroxymethyl group points up, down, up, down. So that means that along a polymer chain, the repeating unit is not really anhydrous glucose, but cellobiols. So we made MD simulation and looked at what's the mobility, or indirectly the relaxation time. You can calculate the relaxation time from the molecular mobility between those two and between the pairs of surfaces. And the outcome of this was that we couldn't see any difference in MD results due to the twofold screw axis. That's along the fibril length. But when you compared different pairs of surfaces belonging to polymers at different Miller indices, or on top of different Miller indexed planes, you can see that the experimental was 18, 5, 11, and the experimental was 7 and 4.8. And that's off by a significant factor, but the ratio of relaxation times was reproduced very well by the MD simulation. So for that reason, we concluded that yes, we have this signal at around 84.3. That's the 1 - 1 0. That's the polymers constituting the surface polymers residing on top of the 1 - 1 0 plane, and the other signal is correspondingly 1 1 0. And this was for temp oxidation, and there was a group later on that made the same kind of measurement with XRD. I showed you this before just to show that changes in supramolecular structure is not only NMR that can see, but you can see that they also noticed that if you go to high degree of substitution tempo, it's the 1 - 1 0 signal that suffers. So this basically confirmed by XRD or wax, which is more or less the definition of these crystallographic planes, that this is the case. And that's also an additional piece of experimental support for this quadratic cross-sectional fibril model that we use. And that was kind of nice. Yes, FSP and pore sizes. Well, when you do measurement in the wet state, you get high resolution and you can determine sizes and whatnot. But the thing is that if you know the density of cellulose, which we do, then you can compute specific surface area. And since the measurements are normally made in the water swollen state, it's the specific surface area in the water swollen state. And there aren't that many techniques that can do that apart from solid state NMR. And you can see that if we were to have individualized fibers, we would have a specific surface area of about 760 square meters per gram. If it's the envelope of the aggregate, and the aggregate is fairly compact, then it's around 130 square meters per gram. So that's the numbers you get basically, and it's in agreement with what has been known since previously by other techniques, that these are reasonable values for what you can expect. But the interesting thing is that the dry fiber has a specific surface area of about a square meter per gram. So swelling it in liquid water means that you increase the specific surface area enormously, at least by a factor of 100 or so. But you can get this from an NMR measurement on cellulose. FSP, the fiber saturation point measurement, is a solute exclusion technique that also deals with the material in a water swollen state. You have a water swollen pulp sample, and then you add a solution containing dextran, and you're keeping track of the total amount of water, and you leave this to equilibrate. And then you, based on what you know of the sample, you calculate the concentration of the dextran, theoretically. And then you take a sample of the liquid in this, you sample the liquid in this pulp water, or the water swollen pulp sample. And then you determine the concentration of the dextran with the optical rotation. You always end up, most of the time, not always, but most of the time, you end up with the concentration of dextran that is higher than the theoretically calculated one. And that means that there is water trapped that cannot participate in diluting the dextran. And from this, you can compute backwards, and you can get the specific volume of water. So you have the specific surface area and you have the specific volume. And if you have a volume per unit mass and you divide it by an area per unit mass, you get the length per unit mass, if you wish. But the point is that this is the thickness that the water layer will have if you smear that water over all the surface area. And that's, as the picture here indicates, that's half the pore size. And we can compute an average pore size of the fiber wall even without having to assume any particular pore shape. So combining these two techniques, since we can combine these two techniques, since they are having the sample in pretty much the same conditions, and the measurement results are immediately comparable, so we can get it from that. And this is of course important when you want to address issues of like enzymatic reactivity and stuff like that. But I won't go much more into detail of that one than just showing you that since we can do all the measurements here in the water swollen state, what we get from NMR in terms of, for example, specific surface area and pore size should be an upper bound to other techniques where you have to, like in the case with BET, where you have to either do a liquid exchange of the sample, or that can deswell it, or you do a drying which can collapse the material so specific surface area and average pore sizes changes. And when we compare it to for 3 different samples where we have both BET and this NMR determinations, it turned out that NMR reasonably well fit the criteria of an upper bound. So that works. Okay, a short thing about the way forward, cryomass. Well, we don't have this technique, but it's a technique that's rather recent. And I was a couple of weeks ago on an inauguration in Umeå where they have bought a cryomass probe, and they can do measurements that speed CP mass carbon-13 NMR up significantly. So the trick is not to cool the sample down, but rather to cool the electronics down to about 20-30 Kelvin. And the sample is still at room temperature, but when you decrease the temperature, or when you lower the temperature of the electronics, you decrease the electronic noise. And that means that you increase the signal to noise, of course. And you can, I mean, historically in NMR, you've done that by increasing the magnetic field strength, which is kind of expensive. Now, the cryomass is not an inexpensive technique, but it's at least expensive than a magnet. So you get improvement of signal to noise. And as you might be aware of, you know, an improvement in signal to noise translate quadratically into a decreased acquisition time. So suddenly, for a moderate improvement, if you can improve your signal to noise by a factor of, say, 10, suddenly you can shrink your acquisition time down by a factor of 100. And I will just show you an example of measurement that we did on the equipment that Broker have. And it wasn't exactly the same thing we have in Stockholm here at RISE, but it's close. I mean, you have a 600 megahertz. This was in Switzerland, and RISE at 400. So there is a bit of signal to noise improvement just from magnetic strength, but that's a factor of about 1.6, which would translate into, I don't know, 2.5 improvements in a decrease in acquisition time. Cryomass measurements were made by their application chemist, Dr. Barbara Peroni at Broker, and we measured at our instrument this is on a difficult sample, which is a rather water-rich, low solid contents, hydrochloric acid CNC made from linters. And we measured this for around 11 hours. We have 16,000 scans, something like that, in order to get the signal to noise over this region, the region where we do the sort of calculations, of around 250. If you do the same measurement on this cryomass probe, same sample, and you reach a signal to noise that's similar, not 250 but 207, but you do this on 250 scans, and it takes you about 11 minutes. So effectively, you drop the acquisition time by a factor of 60, roughly. And that's, of course, something that can increase your throughput enormously. Unfortunately, this instrument, the CryoMass instrument, does not yet have an auto sampler. You have to do that manually, but you can still gain quite a lot of time. Typically, we measure our pulp samples for about 3 hours. That means that we can run, say, 3 samples per day, something like that. And with this, you could run maybe 2 samples per hour at least, or something like that, even with switching samples. So that would be a rather drastic improvement in throughput. And also, since it's the same kind of measurement, it's still a bulk method, it's a CP mass method, and we don't have to mess with the temperature of the sample. You can see that the results we get when we do our sort of spectral fitting analysis, both on the spectra from our instrument and from the one in Germany, we get within experimental or the same results. So it would be a perfect replacement for our equipment if we could get our hands on it. Or if not, we can, of course, use the one that is existing in Umeå. This is within the Swedish NMR Center, so you can apply for time to use it. And with this improvement or decrease, I would say, in acquisition time, suddenly 2-dimensional NMR in solid state becomes much more doable than on a regular room temperature probe. Some final words. I will round this off, and I will come back to this one, that this is the model we have been using for a long time, and several people have tried to falsify that, including us, and we haven't succeeded. And the point here is that we have, this model is built, now it's built on so much experimental support. So if you want to use some kind of model for cellulose isolated from wood, and want to have another model than this one, then I think it is a reasonable, that you should show that the alternate model, you should sort of build this, you should supply the same amount of experimental support for that model as we have done for this model. Otherwise, my recommendation is to stick with this one, because this can explain quite a lot if you use it properly. Secondly, this misunderstanding about cellulose as a class of compounds rather than one compound can be resolved rather easily if you realize that cellulose, of course, from different sources, that was known, but cellulose prior to isolation, isolated cellulose, and chemically modified isolated cellulose, they don't have to be the same. They don't have to have the same supramolecular structure. They can be rather distinct animals, so to speak. And just to show this, 2018, there was a group that did DFT calculation of chemical shift. This is basically quantum chemical calculations. You need that to determine chemical shifts. And they set out to try this idea of whether we have the proper assignment when we talk about signals from the crystalline interior and the surface signals. And they had lots of word here, but eventually they came to the conclusion that this could very well be the case, also based on their calculations. And this was a rather recent one. This was in Cellulose 2018. With those words, I'm done. Thank you very much. And if there are questions, I'll be happy to ask them, answer them. Thank you.
Thank you so much, Tomas. Really nice presentation. And actually, we already directly now have the question in the chat. And you can see it.
Chat. Okay.
Yes, in the chat. Let me see.
I'll try it. You mentioned the cell wall for native cellulose and the isolated cellulose, that they look alike, which does not making sense. How would the cellulose do? No, I didn't. No, I don't get the question. You mentioned about the cell wall for native cellulose and the isolated cellulose, that they look alike. No, they don't. I just said the opposite. They don't look alike. That's the point. And then this follow-up is, how would the cellulose 2 look like? Would the cell wall still remain after dissolution and regeneration process? What is the view on that? I don't know. I know of experiments, but that has been made, but just under, you know, a optical microscope, where they had been using lithium chloride dimethylacetamide as a solution, and you let that sort of seep in between the object glass and the cover glass, and you can, under a microscope, see that a fiber completely disappears. It becomes completely transparent. You can't see it anymore. But as soon as you add a drop of water and let that seep in, the fiber reemerges. You can see that in a microscope. I've been shown that. That was a colleague of mine that did the experiment and showed it to me, how this fiber went away and came back. And see, formally, it could be either really highly swollen or it could be dissolved. In the case it's been dissolved, it's most likely that it came back, the cellulose would have come back as cellulose 2, but if it was only really highly swollen, it could easily come back as cellulose 1 as well. But you can't say really, because the resolution, of course, of an optical microscope isn't sufficient to tell the difference. So is that some kind of answer to this question?
Do we still have the participant together with us? I unfortunately can't read your name. I allowed you to speak. Maybe you would like to get connected and just ask the question directly. In the chat, it says SHA.
Yeah.
I don't know if participant is still with us and listening. Otherwise, if you wish, you can just talk, unmute yourself.
But I mean, if I said, you mentioned that the cell wall for native cellulose and the isolated cellulose, that they look alike, which if I said that, that's wrong. Then I either had sort of said something incorrectly or you have misunderstood me, either way. But that's not the case. What I'm trying to say here is that all these three different kinds of cellulose, if you forget about the source of the cellulose, which of course they are different, but even if you have the same source, prior to isolation, isolated cellulose, and when you do chemical modification of the isolated cellulose, they don't have to have the same supramolecular structure. They can be distinct, and normally they are. So that was the message. And if it's been understood as if I said that they were not different, then that's wrong. Then I expressed myself unclearly.
Mm. But at least to me, now your explanation is very clear. I hope the participant also got the reply to the question.
No.
We move on. We have also a question from Mikael. Mikael, I allowed you to speak. If you wish, you can unmute yourself and even ask the question orally. Otherwise, Tomas, you can read it in the Q&A.
In the Q&A? Okay.
It says, you have mentioned that C4 region is suitable to look at different morphology of the cellulose in the fiber. You can see it now, yeah? Due to isolated C4
yeah, it's kind of simple, because if you try to do it, if you take in the really high, let me put it like this, the simple answer is that in a spectra that is not carbon-13 enriched, you can't do it. It doesn't work. The spectral fitting doesn't work. The spectral fitting is a numerically rather sensitive thing. And if you try to do it in the C1 region, you have so much overlap that you cannot, in a reasonable fashion, distinguish the different contributions. And if you mean the C6 region, the one that is at around 50, 60 ppm, it's the same story. The overlap from 235 makes it extremely difficult. I haven't succeeded in doing that unless the sample has been carbon-13 enriched in C6. C1, you know, all the signals show up in a much narrower spectral range, and they are so overlapped, so it's not, maybe in some cases you can, but it's not very easy. The C4 is, of course, the region, and C4 and C6 is the region where you have surface signals reasonably well separated from the crystalline interior, and that's why they are more attractive. But still, the C1 is difficult, C6 is difficult even without carbon-13 enrichment.
I hope, Mikael, you got the reply to your question. We didn't
Absolutely.
get...
Yes,
yeah, okay.
That I was wondering, but I thought like I can ask these questions here, because as Tomas says, I mean, also C1 is separated region and C6 is separated region, but if different different morphology results in a very poorly separated peaks, then yeah, then I totally understand that the convolution will not work. Thank you.
Thank you so much, Mikkel, for your question. Really good. Gunnar, do we have more questions?
Yes, there is a question about when you sort of compare complementary techniques, like measuring pore size, etc. If you look at sort of time-wise, the techniques you have compared with NMR, would you say that some of them are faster or sort of simpler than NMR, or they are the equal sort of time-wise to make these, like, for example, NMR versus FSP?
Yeah, but I mean, FSP, that's only, FSP gives you just sort of the liquid volume of a fiber wall, so they don't give you that structural information. It's not until you combine FSP with NMR you can get some kind of structural information from that data. But I mean, the clear candidate for comparison would then be wide-angle X-ray scattering, I mean, WAX or XRD. And I mean, measurement-wise, I would say that depends on what kind of equipment you have. If you have a typical lab instrument, you need to do measurement for maybe an hour or so on a cellulose sample, could be a bit more, and then an NMR measurement takes us maybe 3 hours. So there's very little gain really in doing the measurement on wax. And time-wise, and there is a gain, of course, and I think you can normally on lab X-ray equipment, you can automate things so you can measure them more rapidly. But after that, you have normally also a longer process of post-acquisition processing of data when you want to, if you have a 2D detector and you should do the integration and you should do background subtraction and so on. And I mean, none of these techniques are, in my way of looking at them, easy or rapid. They are difficult and they are expensive, and they take quite a while, and we use them not because we are so fond of them, but we use them because there aren't really any other alternatives.
But the conclusion is that stick to NMR, or the first choice should be NMR.
I can put it like this, without trying to be funny, there are a couple of techniques where you can determine the degree of crystallinity of cellulose, which is an issue that comes back for reasons I don't really understand. But the only instrument that is really close to becoming a golden standard for determining the degree of crystallinity of cellulose, in my opinion, is NMR, because that's the method where you are closest, you can determine the degree of crystallinity closest to the measurement data. You don't need a calibration curve, you don't need any substances to compare with, you have NMR has a line shape theory, so you know what kind of mathematical functions to fit to your experimental data, which is not the case really for X-ray diffraction. And you have problems in X-ray diffraction with determining what they call the amorphous content, the amorphous signal, and so on. So all of that is sort of resolved in NMR. And if you can live with the fact that NMR gives you an upper bound for what the crystallinity can be, then that's the best technique.
Thank you very much. Thank you.
Thank you, Gunnar and Tomas. I do not see any more questions. Gunnar, do you see any?
No, there are no more here,
No?
what I can see.
Okay, so we didn't miss any.
Yeah.
So with this, it's our turn to thank you, Tomas. Really great presentation. Yes, thank you so much. And together with Gunnar, we would like to thank all participants and all the speakers. It was a great pleasure for us to organize the NMR webinar series during this year. That's the final webinar for this year.
Okay.
Yes, thank you for the applauses also. Next year, we will continue with the NMR courses. So those of you who haven't signed up yet, please do it. So we have one course already announced on the Treesearch website, and then tentatively more courses will come up connected to NMR also, but that's a little bit later during the year. And Treesearch Infrastructure will be back with more webinars and site visits and other courses. So stay tuned, follow us, and this webinar also will be available soon on our YouTube channel. So thank you so much for today, everyone.
Thank you,
Okay.
Anastasia.
Bye-bye.
So